Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1943 witnessed the publication of a landmark paper that irrevocably altered the course of biology and medicine. Oswald Theodore Avery, along with his colleagues MacLeod and McCarty, presented their meticulous research and studies on the chemical nature of the substance-inducing transformation of pneumococcal types, a work that dared to suggest deoxyribonucleic acid, or DNA, as the carrier of genetic information. This proposition, seemingly simple in retrospect, challenged prevailing scientific dogma and sparked a revolution in our understanding of heredity. Their findings were a monumental step, though one initially met with skepticism, in unravelling the molecular basis of life. Avery's work did exist in a rich historical context. The preceding decades involved intense investigations seeking to define the elusive transforming principle reported by Frederick Griffith in 1948. Griffith demonstrated that a harmless strain of pneumococcus could be transformed into a virulent form by exposure to heat to kill virulent bacteria. This hinted at a transferable substance responsible for heredity, yet its chemical identity remained shrouded in mystery. Scientists pondered whether this substance was protein, a complex molecule then favoured as the genetic material, or some other component of the bacterial cell. Prior work by researchers like Phobos Levine, who elucidated the structure of nucleotides, the building blocks of nucleic acids, provided crucial background, although Levine himself favoured a structural, rather than informational, role for DNA due to its seemingly simple repeating tetranucleotide structure. The work of William Asbury, who used X-ray diffraction to study DNA, further suggested a repeating structure, reinforcing the notion that DNA possessed an adequate complexity to encode the vast diversity of life. Avery, MacLeod, and McCarty embarked on a systematic and rigorous purification process to isolate the transforming principle from a virulent strain of pneumococcus. Their approach began with a lyset of heat-killed virulent pneumococci. They then meticulously removed various cellular components, including proteins, lipids, and polysaccharides, using a combination of enzymatic digestion and chemical fractionation. Crucially, they employed enzymes such as proteases and ribonucleases to specifically degrade proteins and ribonucleic acid, or RNA, respectively. After each purification step, they tested the transforming activity of the remaining material by its ability to convert an unvirulent strain of pneumococcus into a virulent one. The results were astoundingly consistent. As they removed proteins and RNA, the transforming activity persisted and even intensified. However, when they treated the purified extract with deoxyribonuclease, or DNase, an enzyme that specifically degrades DNA, the transforming activity was completely abolished. The evidence pointed unequivocally to DNA as the transforming principle. The team carefully characterized the purified DNA using a variety of biochemical and biophysical techniques. They determined its elemental composition, its viscosity, and its sedimentation properties. Their analyses consistently indicated that the active transforming principle was highly purified DNA, free from detectable amounts of protein or RNA. Further supporting their conclusion, they showed that the transforming activity was highly sensitive to DNase, providing a definitive link between DNA and the phenomenon of transformation. One could consider the experiment akin to identifying a culprit from a list of suspects. The scientists systematically eliminated the proteins, the lipids as polysaccharides, until only DNA remained, bearing the undeniable fingerprints of transformation. The immediate impact, though significant, was initially met with considerable caution within the scientific community. The prevailing belief in proteins as the genetic material, owing to their perceived structural complexity and diversity, cast a shadow over Avery's findings. Many scientists found it difficult to reconcile how a molecule with a seemingly simple repeating structure could possibly encode the vast amount of information necessary for heredity. Furthermore, the experimental evidence, though compelling, was derived from a bacterial system, and some questioned its applicability to higher organisms. Some questioned the purity of the DNA preparations, raising the possibility that trace amounts of protein contaminants could be responsible for the transforming activity. Others suggested that DNA might be acting as a mutagen, causing the non-virulent bacteria to mutate into virulent forms, rather than directly transferring genetic information. Despite initial skepticism, Avery's transformative work gradually gained acceptance and fueled further research. Owing Chargaff's subsequent discovery of the base pairing rules in DNA, showing that the amount of adenine is equal to the amount of thymine and the amount of guanine is equal to the amount of cytosine, provided crucial support for the informational role of DNA. His carefully performed experiments revealed patterns and relationships in the base composition of DNA that proteins simply could exhibit, further undermining the suitability of proteins as the carriers of genetic information. These fundamental laws of DNA composition established by Chargaff were indispensable in the later discovery of the molecular structure of DNA. The work of Alfred Hershey and Martha Chase in 1952 provided irrefutable evidence that DNA, not protein, is the genetic material in bacteriophages, solidifying Avery's conclusions. Hershey and Chase radio-labeled the protein and DNA components of bacteriophages, viruses that infect bacteria, and then allowed the phages to infect bacterial cells. They found that radioactive DNA, but not radioactive protein, entered the bacterial cells during infection, indicating that DNA carries the genetic information necessary for viral replication. This elegant experiment, often referred to as the Blender experiment, finally convinced the broader scientific community of the central role of DNA in heredity. The experiment employed radioactive isotopes of phosphorus to label the radioactive isotopes of sulfur protein. The crowning achievement came in 1953 with James Watson and Francis Crick's elucidation of the double helix structure of DNA. The model, built upon the X-ray diffraction data of Rosalind Franklin and Morris Wilkins, elegantly explained how DNA could both store and replicate genetic information. The double helix structure, with its complementary base pairing, provided a mechanism for accurate replication and transmission of genetic information from one generation to the next. The structure also suggested how genetic information could be encoded in the sequence of the bases adenine, thymine, guanine, and cytosine, paving the way for the development of molecular biology. The initial publication by Watson and Crick was remarkably concise, yet its impact on biology was immense. One might say that they provided the architectural blueprint for the very building blocks of life. The structural nature of DNA was suddenly revealed, offering insights into how genetic information could self-replicate. Avery's study had a profound and lasting influence on subsequent research in genetics and molecular biology. It laid the foundation for our current understanding of gene structure, gene expression, and gene regulation. The discovery that DNA is the genetic material opened up new avenues of research into the molecular mechanisms of heredity, leading to the development of new technologies for manipulating and studying genes. The polymerase chain reaction, or PCR, DNA sequencing, and gene cloning are all direct descendants of Avery's groundbreaking work. These technologies have revolutionized medicine, agriculture, and biotechnology, enabling us to diagnose and treat diseases, develop new crops, and engineer novel biological systems. His work catalyzed the revolution in genetic engineering, allowing scientists to cut, slice, and piece together DNA molecules, leading to a multitude of biotechnological applications. The conceptual shift initiated by Avery's work also had profound implications for our understanding of evolution. The demonstration that DNA carries genetic information provided a molecular basis for natural selection, explaining how genetic variations can arise and be passed on to future generations. This understanding has been essential for understanding the evolution of antibiotic resistance in bacteria, the development of new vaccines, and the genetic basis of human diseases. It provided a molecular explanation for the mechanics of heritable change, allowing scientists to track the processes of evolution at the level of DNA sequence. Despite its revolutionary impact, Avery's work was not without its limitations. The study did not elucidate the molecular mechanisms by which DNA induces transformation. It remained a mystery how DNA could enter bacterial cells and alter their genetic properties. This led to further research into the mechanisms of DNA uptake and integration in bacteria, which eventually revealed the existence of specialized transport systems and enzymes involved in the process of transformation. Later research uncovered that transformation involved the uptake of foreign DNA into the bacterial cell, followed by its integration into the bacterial chromosome through a process of homologous recombination. Furthermore, Avery's work did focus solely on pneumococcus, and it was not immediately clear whether his findings were applicable to all organisms. The subsequent demonstration that DNA is a genetic material in bacteriophages and other organisms provided strong support for the universality of DNA as the carrier of genetic information. It became evident that while the specific mechanisms of DNA replication and gene expression may vary across different organisms, the fundamental principle that DNA carries genetic information holds true for all life forms. Some have argued that Avery and his colleagues should have been awarded the Nobel Prize for their pioneering work. While the reasons for their exclusion remain a subject of debate, it is clear that their contribution to science was monumental. Their meticulous experiments, their unwavering commitment to scientific rigor, and their courage to challenge prevailing dogma paved the way for the molecular revolution that has transformed our understanding of life. Their legacy continues to inspire scientists and researchers to this day. One might say they laid the groundwork for the very edifice, modern molecular biology, an edifice that continues to expand and enrich our understanding of the world around. The story of Avery's work also serves as a reminder of the importance of basic research. His study was not driven by any immediate practical application, yet it led to profound discoveries that have revolutionized medicine and biotechnology. It underscores the importance of curiosity-driven research and the need to support fundamental scientific inquiry. Pure scientific discovery, regardless of any immediate societal need, has the potential to transform. The studies on the chemical nature of the substance-inducing transformation of pneumococcal types stands as a testament to the power of scientific inquiry. It represents a pivotal moment in the history of biology, a moment when the veil was lifted on the molecular basis of heredity. Avery, MacLeod, and McCarty's meticulous work provided the crucial link between DNA and genetic information, forever changing our understanding of life and paving the way for the modern era of molecular biology. The careful experimental design, rigorous analysis, and willingness to challenge established beliefs cemented their place as giants in the scientific world. Theirs remains a story of profound scientific impact, a quiet revolution that continues to reverberate through the halls of science today.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.